Okay. Hello everyone. I'm Sankalpa Chakraborty. Welcome to Disease Biology Interviews. Today I'm here with Dr. Partha Majumder, and he is the Director of National Institute of Biomedical Genomics and Honorary Professor of Jawaharlal Nehru Center for Advanced Scientific Research, India. So, welcome Dr. Majumder. First of all, on behalf of the whole Disease Biology team and our readers, I welcome you on Disease Biology. And thanks for giving us your precious time for this interview. Though as a biologist, you are not bound to any introduction, but first we would like to know about you and your early steps in science, your education, everything. Just a little introduction of you.
All right. So first of all, let me thank you for contacting me and asking me to say a few things, mostly autobiographical, I suppose. All right. So I actually grew up not in Calcutta. I actually grew up in South India in a place called Visakhapatnam. The reason why I grew up there is because my father was a marine biologist, well, actually a hydrobiologist, and he worked for the Central Marine Fisheries Research Institute. The nearest station that they had was in Visakhapatnam, and that's where he worked. And so I was essentially brought up there. I was not born there, but I was brought up there. So I did my schooling in Visakhapatnam, and I essentially grew up there until I migrated to Calcutta to join the Indian Statistical Institute. So I went to St. Aloysius School in Visakhapatnam, which is a French missionary school, and that's where I got my education. I did my pre-university from a college in Visakhapatnam. I didn't know what I was going to do. I mean, those days, none of us actually planned our lives. Unlike you guys, I mean, who exactly know even from grade 9 what you want to do for the rest of your lives. We just took it as a random walk. So the way that I got into the Indian Statistical Institute, and the way that I got into the Indian Statistical Institute was that a friend of my father, a colleague of my father, had done a diploma at the ISI, the Indian Statistical Institute in Calcutta, and he felt that statistics was something that was good. And so he said that, why don't you take the test, admission test, and see if you can pass. It is difficult, but try it. So I was going to become an engineer. In our days, chemical engineering was the big hype. So I was going to become a chemical engineer if I got admission to a degree program, an engineering degree program. But anyway, I took this test in ISI. I didn't know anything about statistics or even what statistics was. I had mathematics, physics, and chemistry in my specialization in high school and pre-university. And so anyway, I passed the test, and so I said, well, let me go and try it out. And essentially, again, a series of random walks, random events, I landed up at the Indian Statistical Institute, and that's something that I always think that, that has stood me in good stead. I think that was a fantastic thing that happened, and so I joined the Indian Statistical Institute in their Bachelor of Statistics honors course in 1970. The ISI is somebody who enters in BStat rarely ever goes out after doing a bachelor's, so we continue on to do masters. Our bachelor's program is 4 years, master's program was 1 year. Things have changed now. At that time, when we graduated, our bachelor's program was 4 years program, and therefore nobody would leave ISI without doing another 1 year of master's. So that's what I did. We got a very wholesome training at the ISI, and that was really nice, both applications and theory, a lot of mathematics, a lot of statistical applications as we went forward. I had a fantastic set of teachers at the ISI who were also world leaders in their own areas of research. So all in all, it was fantastic. I actually got to see even Prasanth Chandra Mahalanobis, the person who founded the Indian Statistical Institute and did the second 5-year plan of India. So So Malanobis came and took one class for us when we were in the BStat second year, but passed away shortly thereafter. So all in all, it was a fantastic experience. I graduated with applied statistics and data analysis as my specialization in masters, and then went on to do a PhD from the Indian Statistical Institute in biostatistics. For my PhD, I was essentially left on my own. By then, there were hardly any biostatisticians in ISI, but the person who was my supervisor knew a little bit of biostatistics, primarily because he was, he was already a faculty member when Professor C.R. Rao was the director of ISI, and J.B.S. Haldane was also a faculty member of the ISI. So that's how he got his initiation into whatever little biostatistics he knew. So anyway, I did a PhD in biostatistics, and then moved on to do population genetics. I went to the University of Texas Health Science Center in a small center called the Center for Demographic and Population Genetics. So that's where I did my postdoctoral training. Then I spent a few more years later at the University of Pittsburgh, and essentially joined the Indian Statistical Institute as a faculty member. And in 2010, when I mooted this idea of forming this new institute called the National Institute of Biomedical Genomics, I'm still a professor at the Indian Statistical Institute. I'm on deputation, on long-term leave from ISI, and I'm trying to set up this institute. So that's what my life course has been.
I mean, that's great. I mean, now it's clear why, I mean, you are basically a statistician by education, and now that during your PhD you moved to biostatistics part, as far as I can understand.
So I actually had some interest in biology early on. My father, as I said, was a hydrobiologist, and so I guess I got it's mostly environmental influence. So at home, the environment was conducive to thinking about science, and so I got excited in science. So I think the seeds were sown because of the environment at home, primarily because of my father. And so I was interested in biology, and I started reading biology, did a PhD in biostatistics, but the kind of biology that I do today and understand today was only possible because I did a postdoc in the US, and more than doing a postdoc in the US, I had a deal with my postdoctoral mentor, and he was very kind and allowed me to take essentially goof off during the daytime and do the postdoc work in the evenings. So what did I do during the daytime? So I went and took courses, starting from cell biology to molecular biology to molecular genetics, biochemistry. We didn't have these courses at the ISI, even though we did have some basic fundamental courses in biology, but we didn't have these deeper courses. So I went to the UT Health Science Center courses. I went to Rice University courses. So this is what I would do during the daytime: take courses and execute my responsibilities as a postdoctoral fellow later in the evening. So they were extended periods of time. Also had a very good friend, a colleague who is a little bit senior to me, but he's a biochemist. Not only taught me biochemistry, but what he did was something phenomenal. He allowed me to get into his lab and break, pipettes and deal with pipette men and, blow up electrophoresis gel chambers and stuff like that. So he allowed me to be a gorilla in his lab, and that really helped me. I did hands-on work, at that time, RFLPs, electrophoresis, all those kinds of hands-on work I learned in his lab, the PCR and so on and so forth. So aside from taking these courses, because I was allowed to be a gorilla in his lab, I think it really helped. So that's how I learned biology. The interest in biology was kindled long ago, but to be able to learn biology formally by taking biochemistry courses, cell biology courses, etc., all of that came during the 3 years I was in Houston doing a postdoc.
Oh, that's great. I mean, currently, what is your research focus in your lab, and tell us something about your research.
So my research has been quite varied, quite varied as a matter of fact. Like you said, you mentioned a couple of times that I'm essentially a statistician. Yes, I am a statistician, and the reason why I believe that statistics and biology are so well enmeshed. Today, it's almost in every sphere of biology needs statistics. But when we were starting, the area of biology where statistics was most applicable or applied is genetics. And in genetics, human genetics was more attractive primarily because you couldn't do crosses. When you could make crosses of homozygous cell lines, let's say opposite, not cell lines, homozygous parents, you could predict what the offspring will be using Mendel's laws. But in humans, you couldn't make crosses, deliberate crosses. So it was a little bit more statistical intricacy, and that was very attractive at that time. We all grew up at the ISI with a lot of quantitative thinking, and that quantitative thinking permeates biology to a great extent. So that's what my training was. But essentially what I did at later points of time, I divided my time in three areas. One is I was very interested in statistics, development of methods of statistical analysis of biological data. So I spent a significant amount of time doing that, and that was also a part of my PhD thesis. Or those kinds of things were also a part of my PhD thesis. The second area, as we moved along, that I got interested in, and I got interested in again because I was at the Indian Statistical Institute, is that the Indian Statistical Institute had a fairly active and not very large but fairly active anthropology unit where they looked at anthropological diversity of the people of India. And so initially it would be anthropometry, body measurements, etc. But over a period of time, they started collecting blood samples and looking at that time, looking at essentially biochemical markers. So they were trying to understand what the relationships among these various ethnic groups were. And when I got fascinated by those kinds of things, so I spent a considerable amount of time as, first of all, my repertoire of knowledge increased. Second of all, technologies have increased, so we moved on to use more and more deeper technologies, more informative technologies in order to generate data that would essentially ask the same kinds of questions. Where do we come from? And because I was in India, I obviously was asking questions like, how was India people? Why do we have so much of ethnic diversity in India, and how do these ethnic people relate to each other, and what kind of migrations could you even trace migrations using genetic loci and genetic data? So reconstruction of human evolution, understanding peopling of India has occupied a large area of my research interest. Development of statistical methods was the second area of interest. The third area of interest was kind of very broad, like your website is also called disease biology. So I would say that broad area of work was called, can be called as disease genetics. So I was dealing with, so I was not, I've never been tied to one single disease.
Okay.
And as I analyze my own life, the area of work in, or the kind of work that I've done in disease genetics, again had to do with understanding the methods that are applicable to those kinds of questions, evolving simultaneously methodologies to analyze those kinds of data. We started our lives with monogenic disorders. Quickly we moved on to more complex disorders where the methods of analysis that are used for monogenic disorders were not applicable. So over a period of time, I sort of dabbled on very many diseases. Again, no very deep contributions to the understanding the biology of a disease, but certainly what we did was to understand the genetics of the disease. Sometimes they were familial. Why was the familiarity caused? Sometimes they were sporadic, meaning is there not a genetic underpinning to that? All that I will say is, what have I been doing in the last 5 years? I've been very focused, concentrating on a specific project or a specific subject area, which is oral cancer. And this again was serendipity. I mean, it was not like I can again trace back my interest to my postdoc days. So let me tell you a little bit about the fantastic kinds of people that I came in contact with when I was doing my postdoc. So I don't know if you have heard the name of Alfred Knudsen. Al Knudsen, the last name is spelled as Knudson. First name is Alfred. You're too young. You may not know his name, but Al Knudsen.
Yeah, may I
You know his name?
I mean, Alfred Knudsen, he phrased the two-hit hypothesis.
Correct.
Knudsen, okay.
So, absolutely. So you're completely right. So Al essentially, provided the theory or the reasoning behind his proposition of the two-hit hypothesis. The two-hit hypothesis has been expanded to a multi-hit hypothesis, etc. But the essential components of the argumentation remains. And Al Knudsen was the dean at the Graduate School of Biomedical Sciences at the University of Texas. And he had moved to Chicago, right? Yeah, I think Chicago. And soon after I joined, but he would come back every so often. And when I was doing my postdoc, I was working on various kinds of, again, statistical methods as related to cancer research. Two big-time oncologists in Houston. One person's name is Louise Strong, and the other person's name is Dave Anderson. Dave is no more, but Louise is still alive and active. So I was working with them, and Al Knudsen was actually looking at retinoblastoma, and Louise Strong was the person who was the doctor who was actually looking at retinoblastoma. So it was the combination of Louise Strong's work and Al Knudsen's foresight and argumentative and reasoning capabilities, because of which he was able to propound the two-hit model. And I was doing my postdoc at that time, and we had, I can recall so many interesting meetings with that group: Dave Anderson, Louise Strong, Al Knudsen, and so it has very fond memories. So that's when I started my cancer work. Today I work on oral cancer again. Essentially trying to understand what the genomic or the mutational landscape is. At that time when we were dealing with retinoblastoma, that's I started my life with retinoblastoma, or life in cancer with retinoblastoma. It was kind of familiar. There were sporadic cases, and that's how the two-hit model came about. But today we know that all of cancer is essentially genetic. At the cellular level, it's completely genetic. Even sporadic cancers have actually cellular alterations in the DNA of the cells and so on. So it's, in some ways, it's completely genetic. So that's attractive. So right now what we are trying to do is to look at oral cancer and look at the mutation landscape in oral cancer, essentially somatic and germline, but germlines are only about 5% of cases. It's mostly sporadic. We are also trying to understand what kind of environmental exposures provide the susceptibility or enhancement of susceptibility to oral cancer. So this is what I've been doing in the last few years. Again, this also came kind of all of a sudden, just as the institute came all of a sudden, the new institute. Well, it didn't come all of a sudden. It took a long time to polish thoughts and propose, etc. But still, similarly, the International Cancer Genome Consortium project also came somewhat suddenly, and so India didn't participate in the Human Genome Project. India didn't participate in the HapMap project, and this was the third major international collaboration that came about. I, as a human geneticist, was sad that India did not participate in the Human Genome Project, did not participate in the HapMap project. So this was a third chance, and I sort of went all out in order to contact the administrators, the decision makers, etc. And cutting a long story short, it happened. And I was also asked to coordinate this. So we are right now working with clinical, the most famous clinical entity in India, cancer clinical entity in India, which is the Tata Memorial Hospital, and us. So we do all of the genomics and the statistical analysis. They do all of the clinical analysis, the biospecimen collection, recruitment of patients, and so on. So it's been a very fruitful collaboration. We've had some quite a number of good publications and interesting inferences. So that's what I've been doing in the last few years.
Yeah, I mean, that's great. So, I mean, in your opinion, how much successful this International Cancer Consortium is, Cancer Genetics Consortium is till date?
So I don't know what your measures of success are. I don't know. I really don't know how to measure success in science. But anyway, what the International Cancer Genome Consortium has been able to do is a large number of cancers. First of all, they've been able to um, make scientists of different nations come together. That itself is something that's wonderful in science, because if you're going to do big science and that needs a large number of samples, deep technologies, generates massive data sets that need multiple heads to analyze. So coming in the consortium mode is extremely important and interesting, and bringing in a whole bunch of people dispersed in different geographical regions of the world, bringing that together is itself a measure of success, I would think. So that was their first initial success. In terms of the scientific successes, so different countries have been, or scientists in different countries who are under the umbrella of the consortium are working on different kinds of cancers. We in India are working on oral cancers, primarily because it's very prevalent in India. It also has certain epidemiological features that are different from the ones that you find in the West. So there is a major attraction towards understanding the so-called Indian variety of oral cancer, which is the oral cancer in the gingiva-buccal complex, essentially the oral cavity. Most of the oral cancers that you see in the West are tongue cancers. So we don't see as many tongue cancers, we see more epithelial cancers of the oral cavity. So that's what makes it an Indian problem. So we are interested in that. So as I said, different countries have now identified the mutational landscapes of all of these various types of cancers, over 50 different cancer types. The sample sizes are good now. It used uniform set of instrumentation, in more or less uniform homogeneous sets of analytical protocols. So that was good. Harmonized data sets are very, very useful.
So, like at this time point, we are all almost aware that the cancer is becoming more severe due to the fact of the drug resistance.
Mhm.
So, in your opinion, how much personalized medicine, I mean, how much it's probable for the patients, cancer patients right now?
So I can't make predictions. I mean, it's very difficult to make predictions. What I can do is to narrate to you. Let's say, let me just stick to one. So if you look at melanoma, let's say, or it also happens in the same mutations also take place in a large number of non-small cell lung carcinoma patients. So if you look at BRAF, so there are mutations in BRAF that are strongly associated with. To begin with, it was only an association. Now we know causality as well. So this BRAF, we know what it does. It essentially controls signals from outside of the nucleus to inside of the nucleus, and these the signals tell the cells to grow or not to grow, to stop growing, and so on and so forth. So essentially, it's controlling signals from outside of the nucleus to inside of the nucleus, and that's what the role of BRAF is. It tells the cells to stop growing, and that's one of the things that is very, very important, that you have to have regulated cell growth. Dysregulated or unregulated cell growth is the hallmark of cancer. And so what happens, and this is only one of the genes, but one of the major genes, the BRAF gene gets mutated. And the mutated protein then does not perform its normal function, which is to tell the cells to send signals from outside of the cell to inside, telling it to stop growing. So the task that it is supposed to perform goes haywire. So what you really want to do is, when you have a mutation, when you have a mutant BRAF, you want to somehow block the impact of that or effect of that, or make it stop from producing the mutated protein, because the mutated protein then does not function normally. But you don't want to deal with, you don't want to tamper with the wild type protein. The wild type protein is good because that's sending the appropriate signals. So the point is that identifying this mutation or these mutations in BRAF, and there's one predominant mutation in BRAF, that predominant mutation, there are also other mutations. So the idea is that could you then impact on this one mutation? So if you take an individual, if you say that this individual has this mutation, could you then take a chemical molecule and impact on this one mutation? That has actually been done. So we now have a drug in the market, not in the market, it's actually in used in clinical settings. It's called vemurafenib. Essentially, it targets this one single mutation that changes that causes a change in amino acid and leads to the mutated protein. So this particular drug has an impact on survival of the patient. So we know that we can now, having knowledge of these mutations has enabled us to understand what these mutations do, has enabled us to understand the biology, has enabled us to understand chemical molecules that will only impact on the mutated protein or impact on the mutation, not on the wild type. And then we know that it also impacts on survival. So the whole gamut, that starting from identification of the mutation to understanding the biology to getting a drug and seeing the impact of the drug in patients, this whole thing is made possible because of these studies of mutational landscapes, etc. Do we have many examples? We actually don't. But it's too early days because we are still trying to understand the mutation profiles, etc. And so it's too early days, but there are good examples, and we believe that this understanding will lead to precision medicine or personalized medicine.
Okay, that seems hopeful at least. So what do you think currently, what are the lacks in this field of research? You have been in this field of research for some time, and what do you think? What should be
So I
done?
don't know. I mean, I can't pinpoint any shortcoming in this field of research because we're all doing the same thing. Some of us are able to do it faster than others, but this kind of field needs a lot of different kinds of experimentation on cell lines, on animal models, and so on and so forth. So technologies are also improving. So right now we can look at impacts on the genome-wide scale. We have heard about CRISPR-Cas9 technologies that are now very, very useful in order to figure out causalities, functional causalities, or functional validation of the mutations that we identify using DNA sequencing or even RNA sequencing, and so on and so forth. So I don't see any major shortcoming except that funds are limited, skilled manpower is limited, and so necessarily it's got to be baby steps. But these baby steps are becoming adult steps, and so one's hopeful that one will be able to do it. The other major problem is the interactions among the various proteins in the human body. In a petri dish, you have a substantially smaller number of proteins perhaps expressing themselves, and so what looks fantastic on the petri dish, once you translate it to the human, you suddenly find that it doesn't work. And one main reason why it doesn't work is that you are dealing with a smaller, a less diverse system in the petri dish or even in an animal model. You mentioned about Drosophila being used as a model for cancer, but Drosophila is not human. And what we call as a model is in the mind of the beholder in some ways. Obviously, it has to do with evolutionary past, our evolutionary past compared to Drosophila, but still a Drosophila is a Drosophila and a human is a human. And what we mean by model is, again, somewhat in our own mental construct. So this has been one major limitation, that we find some very exciting things in the petri dish or in the animal models. And when we move it to the human, it's somehow the failure rate is very high. And moving into the human is not easy because you just really cannot play with human lives. So there are more and more constraints that are being set up for right reasons in trying to do clinical trials, trying to validate different kinds of molecules and so on. So those are, I wouldn't say shortcomings, but people are becoming more and more cautious, careful, and that is certainly retarding the pace at which we can move. But I'm not complaining. I mean, obviously we need to be cautious, and so even if it means that there'll be a deceleration of pace of moving forward, even so, I mean, we need to be cautious. So these are the various kinds of things that are happening.
Yeah. Okay, so coming back to you, so what do you consider as your biggest achievement as a or as a scientist, as a researcher?
Oh, I have no. It's all been baby steps. I've not made any major strides in my own research or in impacted on some other scientists' lives. No, this question is irrelevant so far as I'm concerned. No, it's been a continuous period of excitement, of struggle, and so whatever I've been able to achieve is just a small baby steps.
Okay, so for the young researchers, sometimes biology can be synonymous to frustration and failures. If you were in research, at least, then what would be your advice to the young researchers as well as students for a successful research career?
Well, first of all, not to be frustrated. That's the first thing that I would say. There are failures in life all the time, and if failures get you, then you can't move forward. Don't let failures get you. That's the most important thing. Can you completely avoid failures? The answer is no. There will be failures in whatever you do. So the one way to avoid failures is not to do anything. That's the best way to avoid failures, and that's the last thing that you would want to do. So be excited about science. Take it as it comes. There will be failures. There will be successes. Be excited about successes. Don't be depressed about the failures. Science is incremental, and necessarily that's a body of knowledge. It's a solid body of knowledge. It's incremental. Just remember that by doing science, you are contributing to this body of knowledge that will be able to move science forward. And that's the conviction that you should have, and work hard and just be at it. I mean, it's very, very exciting. Every day you learn new things, and essentially you're working for yourself. You're not working for anyone else. So that's the excitement in science, and just keep the excitement alive. I think that's the most important part.
Oh, that was really motivating. And the last question for this interview. So, what is your opinion and suggestions about initiatives like This is Biology? And we are trying
Well,
something.
initiative meaning the what you are trying to do with this website, with the interviews and so on and so forth. Yeah, I think these are important, and science is not devoid of people who do science. And young researchers, when I was young, I got inspiration. I mentioned about Al Knudsen. I can tell you stories about him. The way that his mind worked. It's important to understand how people's minds work, and you can only understand how the minds of scientists work by talking to the scientists. So I think these are interesting initiatives that you're taking. You have to obviously choose a large number of people who are at various stages of their lives, because each person will have a different say on how she or he will want to project science to the other people. So I would say that don't just restrict yourself to the bald-headed people like me, but also get to the hairy folks like yourself, younger and with lots of hair and bubbling with enthusiasm. So that's very important, and do make these kinds of things available. Do make other resources available, what I would say is that one way to move science forward is to network, and I'm not using the word network in the context in which network is being used, like in the Facebook and so on and so forth. I'm talking about something a little bit more serious than that. Serious reading, serious failures, like you mentioned about failures. If you have a network and if you talk to different people that, look, I mean, I was doing this, I failed. Somebody might have an interesting take and say that, you know why you failed? I have faced the same thing and this is what I've done to avoid recurrence of the failure. So this kind of, talking amongst each other really helps. And if there are bald-headed people like me listening in, then maybe we can also share some experience with you, and that's a good thing to do.
Oh, okay. So, yeah, it's been really good to talk to you today, and thanks for being here and just sharing your experience and everything with us. So, yeah, thank you again.
Yeah, thank you. Thank you for having me over, and it's a pleasure. Yeah,
Okay, okay,
thanks.
okay. So,
Okay, all right. Thank you very much.
thank you.
Bye.